Hello, welcome to the sixth lecture of this course. My name is Xu Tan, and I'm with the School of Pharmaceutical Sciences of Tsinghua University. Today, I'm going to introduce to you the interactions between viruses and their hosts. We as human beings have lived with viruses for many years in the scale of tens of millions of years. Viruses are the master of the game of the numbers. These tiny creatures are actually the most popular biological entities on Earth, thanks to its tiny size in the range of nanometers. It has been estimated that there are 10 to the 31st virus particles on Earth. And a healthy person, on average, harbors over 10 kinds of chronic viruses. In human feces, per gram, human feces contains estimated 10 to the 9th viral particles, which is quite amazing as you think about it. But these viral particles are likely harmless since we have co-evolved with us for a long time. So we have come to terms with these viruses. It can tolerate each other. In fact, in our genome, there are estimated 8% of so-called endogenous retroviruses. These are viruses that have infected us during human evolution, and now they have been integrated in our genome, becoming part of us. This shows that viruses and humans have really co-evolved for a long period of time, and this co-evolution is often likened to as the so-called red queen hypothesis. This hypothesis was taken from a popular fairy tale written by the British writer Lewis Carroll called The Looking Glass. In this fairy tale, a little girl named Alice walked into a world behind a mirror where she met a queen who is red all over. So this red queen, as you can see, then grabbed this girl to run with her. The red queen told Alice, in this world, it takes all the running you can do to keep in the same place. So people have used this metaphor to illustrate that both viruses and their host have continuously evolved. So they are adapted to each other to maintain this broad coincidence in relative fitness. In another popular analogy is that there is a constant arms race between viruses and their hosts. So both the red queen hypothesis and the arms race analogy highlight the co-evolving relationship between viruses and the host, which can guide us to study the virus-host interaction. So my following lectures can be divided into three parts. First, I'm going to talk about the dependence of virus infection on host cells. Viruses rely on the cellular machinery to replicate. So recent studies have really revealed hundreds of human genes as key viral host factors. So I call this the receiving part of this lecture, basically how our cells receive or enable viral infection. Second part, I'm going to talk about the antiviral defense of human cells, which can classify into innate immune defenses and adaptive immune defenses to try to contain the viral infection. This is what I call the resisting part of this relationship. The third part, I'm going to touch on the drug discoveries based on the viral dependence as well as the immune responses. Basically, I call this the resetting part of this relationship. We try to reset or even to contain or even stop the viral infection and the related pathogenesis. So first, I'm going to talk about the receiving part, how viruses build their life cycle based on human hosts. Here, first, let's delve into a cell and take a look at how a virus infect and replicate itself. So here I'm using SARS virus as an example. So COVID-19, since it's less well studied, but its life cycle is believed to be quite similar to SARS. So as you can see, the viral particle of both SARS and COVID-19, they are characterized by an outer layer of proteins in the shape of baseball bats. So overall looking, these particles resemble a crown. That's how coronavirus got its names. So the spike protein sticking out from the viral particle, and that protein is a key for the attachment of the virus to the cell, host cell, to the surface of the cell that they're going to infect. So-called spike protein, this baseball bat-like protein of the viruses, bind to a cell surface protein named angiotensin converting enzyme 2, or ACE2. And this binding then promote the engulfing, or the so-called endocytosis of the viral particle by the cells. Basically, this cellular membrane basically invaginate to form a bubble. This is so-called the endosome, which enwraps the viral particle inside. After being engulfed, the virus membrane is somehow fused with the endosome membrane, which then opens up a hole so that the RNA genome of the coronavirus can be released into the cells. Coronaviruses are positive sense single-stranded RNA virus. So, which means that their RNA genome can be used as a messenger RNA for protein translation. Also, this RNA is the template for its genome replication. So with the help of the so-called RNA-dependent RNA polymerase of the virus, encoded by the virus, the positive sense RNA is first transcribed into a negative sense RNA, is again transcribed into positive sense genomic RNA, as well as other messenger RNAs, thereby replicating the RNA genome of the virus. And this messenger RNAs are then translated with the help of host cell ribosome into new viral proteins. And then these new viral proteins are processed and assembled with the new RNA genome to generate new viral particles, which are subsequently released from the cells through a process that's like the reverse of endocytosis. Basically, they form a bubble inside, and then this bubble is released outside of the cell so that the viral particle can get out of the cell and initiate another round of the infection. As I mentioned, the first step of the virus life cycle is the binding of the spike protein to the ACE2 receptor. Here, I'm showing you this crystal structure of the so-called receptor binding domain of the spike protein, basically the part of the virus spike protein that is responsible for binding to its host cell receptor. So basically, you can think of the receptor were first the receiving dock of this virus particle. As shown, the receptor is shown in green here. As you can see, the viral spike protein have extensive interaction with its receptor, providing a high affinity binding between these two proteins, which is really key to the entry of the virus into the cells. And given this is the first step of the life cycle, this binding has been extensively studied, and it is also the target of many potential antiviral therapies, such as you heard, neutralizing antibodies or even other soluble proteins, which are aimed at disrupting this interaction so that the viruses can no longer enter the cells, stop the infection process at the very beginning. So after this binding, other than the ACE2 protein, the receptor, are other human proteins, also called host factors, that are involved in this process. A recent study shown that for COVID-19, or the virus also called SARS-CoV-2, just like SARS virus, a human protease called temperase, which also is a membrane protein sitting on the surface of the cell membrane. It's actually very important for the activation of the spike protein. Basically, it digests the viral protein to activate it, viral spike protein to activate it. It changes into a conformation or get into the right shape so that it can bind to the ACE2 receptor. As you can imagine, this protease can then be targeted by some protease inhibitor. Since protease, there have been many drugs that have been developed to target proteases. Actually, it has been shown that protease inhibitors can block the activity of this temperase enzyme, can actually inhibit the entry of the COVID-19 into the cells. So this is another example that we can target this entry process to inhibit viral infection. As I just mentioned, after this binding between the spike protein to the receptor, which is the first engaging activity between the virus and the cells, then this binding then triggers the endocytosis. The cellular membrane then invaginated to form this bubble, which wraps the viral particle inside. This bubble then migrates through the cytoplasm, where it gradually got acidified. But basically, its pH is decreased gradually, and this decrease of pH then triggers the fusion of the viral membrane with the endosome membrane, which then release the viral genome into the cytoplasm. That's how the virus enters the cells. So this is for enveloped virions, such as coronavirus and most of the virus. But there are also non-enveloped virions, which then enter the cell through either lysis of the endosome or permeabilization of the endosome membrane. Eventually, the viral genome got into the cells to start the next process, which become really complicated after the entry step. Actually, recent genome-wide RNA screening have uncovered hundreds of human genes, in this case for HIV replication. RNA screening is that we use the genomic tool called RNA interference. People can screen human gene one at a time to ask the question, how many genes are really required for the virus infection and replication? So based on a large group of studies, this paper really summarize the many human host factors or proteins that are required for the viral replication. As you can see, that the endocytosis and nuclear import, how the HIV DNA got into the nuclease, and the integration of the viral genome into the host chromosome, as well as the transcription, mRNA processing, and the export of RNA from the nucleus back into the cytoplasm, and protein translation, and as well as release, really requires a cohort of human protein to accomplish this process. as you can imagine, if we can use genetic perturbation to inhibit these genes, and that eventually lead to inhibition of viral replication. So this really tells us if you can somehow use small molecule drugs to block one of these genes, you could, in the same vein of reasoning, you could block viral replication. So this understanding really provided us with many possibilities to new possibilities and new ideas to develop new antiviral drugs.